Um, can you all see the screen, like the presentation screen? Okay. So yeah, thank you for the opportunity to share our work with you. So there's me and Sam is on the call as well. He's my supervisor. He's a computational scientist at Argonne. So we're both part of the KBase Model Seed project, but Sam has worked more extensively on the Model Seed section that's concerned with plant modeling. So that's called PlantSEED. And my role was to bring in the machine learning modeling to the metabolic modeling that is already being conducted here at within the project. So today I'm going to be talking about laying the groundwork for applying machine learning to metabolic modeling and how we can use that to integrate different parts, including cofactor or micronutrients within the simulation of metabolic networks. So I'll be talking about why we want to do this, and then the methodology of really how we can do it in, just one way in which we can incorporate that. And so I'm going to talk about two main aspects of this pipeline that we've been working on, which is the biologically feasible flux and the hybrid mechanistic machine learning simulation. And then I'll give an example of a project that we've been part of, from which we could get some data and then experiment with this pipeline. So I'm going to start from the bottom and just say, we really want to do multi-omics integration. Let's say we have the Calvin cycle. I know this is the pathway, these are the reactions, but how can I integrate multiple layers of data with this pathway so that I can get a holistic understanding of what's going on in the metabolic network? So what we want to eventually do is be able to say, oh, here are some of the enzymes that are dependent on these cofactors. So let's say, for example, I know that there's enzymes that bind zinc and enzymes that bind iron, and I can pinpoint where in the pathway I am dependent on these cofactors. Then I want to integrate another layer and say, of this pathway, here are the enzymes that under certain growth condition are significantly up-regulated or down-regulated compared to a control condition. So I'm going to say for these certain reactions, these are the enzymes, and I know which of these enzymes bind zinc and iron, and so I can understand how zinc and iron treatments affect these enzymes. But I want to go one step further, and I'm not satisfied with just looking at enzymes, but I want to look at enzyme complexes, because an enzyme complex is really what catalyzes the reactions. And so I want to know at a reaction level, how is that data affecting the reaction itself, in and of itself. And so that's where we use what we call reaction expression scores, and I'm going to talk about that later on. To see how the reaction is either highly expressed or has a lower expression compared to a control condition. And then finally, I want to know how these reactions affect each other, because they are in a network. And so based on the reaction expression score, I want to know how they are interconnected and how they affect each other, and how, for example, the expression of that reaction is correlated or not correlated with how the enzymes are up or down regulated, or how an enzyme complex a score reflects on the network. So to be able to do this last part, which is reaction fluxes, we need to simulate the network. And one way in which we've been simulating the network is using flux balance analysis. I don't know how many people here are familiar with it, but you take a metabolic network and I look at how reactions are related to metabolites. And so metabolite 1 plus 2 metabolite 2 will produce 3 of metabolite 3, for example, from reaction 1. So based on this metabolic reconstruction, and we generate the stoichiometric matrix, which just tells me, it's a matrix that just tells us how each reaction is related to each metabolite. And so if there's a negative number or stoichiometry, then it means that it's being that the metabolite is being used by the reaction, and then if it's positive, then it's being produced by the reaction. And then we represent this as an integer linear programming model that has some certain constraints that are related to macronutrients. So then I have an upper bound on the uptake of CO2, for example, or the uptake of water or of light. Whatever is the limiting factor here, then we can have some upper bounds on that. And then I simulate the network at steady state, which means whatever comes into the metabolic network has to come out. So if I get so the uptake of carbon, for example, has to be equal to whatever is biosynthesized in terms of carbon. And then we try to maximize the biomass. So the biomass will have a certain number of metabolites or components. And then we try to maximize the production of these metabolites at steady state. Now, this has been working great, and there's a lot of variations to FBA, but then the problem with it is that it's linear. So any change to any of the reactions is only linearly reflected in the biomass. So it doesn't really allow for a lot of variability in terms of changing the growth conditions. And also it doesn't integrate the data sets that actually reflect the change in growth conditions. And it's too constrained. It's really restrictive. Now, what if we just use machine learning? So I'll take one off-the-shelf machine learning methods and I apply it to this, and I will need a lot of experimental data. Since you're working on plant science, you do know that there's very limited data sets, especially open source data sets with, let's say, transcriptome and fluxome or transcriptome and metabolome that all come from the same experimental setup and from the same tissue, from the same time point. We've been actually having some issues with finding those data sets. And to train a machine learning model, you need a really large data set. So then the other problem with that is that it's a black box, so it's hard to interpret what's going on. So then what if we find a method that actually is a compromise between the two, but is actually much better because we don't need a lot of data, but we also can have more interpretability and we can remove that linearity of FBA. So this method is known as physics-informed machine learning. And it basically, what it does is it takes it models the problem as ordinary differential equations, and it uses these equations to actually within the machine learning model itself. So then we can take, in our example, we can take kinetics, transcriptomics, cofactor information, or really any constraints or any information about the metabolic network that can be used as constraints and integrate those within the machine learning model. And I'm going to talk about this later on. The advantage of this is that you can better understand the predictions, but you also need a much a smaller data set for training or for optimization. So how can we do this? We've been working on a pipeline where we take experimental data set. We can plug in here any project or data from any project, as long as we have at least a transcriptome. But if you have a transcriptome and a metabolome or transcriptome and fluxome, it's much better. But the least you can take is a transcriptome, and here we use it as a TMM. And then we create or generate the metabolic reconstruction. And in this example, I'm going to be talking about just a plastidial metabolic network, not the full primary metabolic network. So we start from the genome. We use the apps from KBase that I'll talk about in a bit. And then we implemented a separate app where you can take the primary metabolic network and extract the plastidial model. And we have one for leaf and one for root because the leaf can use light, but the root cannot. And then we use this metabolic network and the transcriptome to compute the biologically feasible fluxes. And then from there, we use those for the machine learning simulation. I'm going to go through each of these steps in the next slides. So how do we use KBase and ModelSEED? There's two main apps that we've used, and there's a lot of apps on KBase that you might be interested in using. But the one that we use is the annotate plant enzyme with OrthoFinder. So what this does is it takes a PlantSEED manual curation of Arabidopsis, and it tries to find the orthologs in the plants that we've been working on are poplar and sorghum. And because there's an implied homology between poplar, sorghum, and Arabidopsis, they will have the same metabolic network, but the genome is just going to be the difference between these. So OrthoFinder finds the orthologs and it helps us annotate the genomes of poplar and sorghum. And then we use reconstruct plant metabolism. So once we have this annotated genome, we can reconstruct the model with the annotated genome, compartmentalization, and associations between genes and reactions. And these are really important in integrating data that is specific to genes to actually an enzyme complex and hence to reactions. These apps are readily available on KBase, the platform, but they've been published in the papers that are cited here, but we've been adding a lot of enhancement to these, and that will be published sometime soon. So after we create the metabolic model, or the metabolic reconstruction, we want to integrate the dataset or the transcriptome with this dataset. So what we do is we compute what's called the biologically feasible fluxes. And the first step in doing that is to compute reaction expression scores. So that's how much a reaction can be, or we also call it the active enzyme abundance, because the abundance of active enzymes is what tells us how much of the catalysis of the reaction will happen or in the simulation. So we take TMM data, the plastidial model, and we compute reaction expression scores as follows. So this is a hypothetical reaction with 3 enzymes. So let's say this reaction can be catalyzed by one of these 3 enzymes. Each enzyme is typically composed of a number of subunits, could be 1, 2, 3, or more. And each subunit has a certain number of paralogs that can constitute that subunit. So then we say, because each subunit can be composed of any of its paralogs, then the abundance of this subunit is the sum of the abundances of the paralogs, because any of them can be used to form the subunit. But then because an enzyme needs all of its subunits to be fully functional, then the abundance of this enzyme is basically limited by the subunit that has the lowest abundance. And in this case, we take the lower abundance of each enzyme, so for a subunit, sorry. So for enzyme 1, it's subunit 1, 2, and so on. And then because the reaction can be catalyzed by any of the 3 enzymes, its score is just going to be the sum of the abundances of each of these enzymes. And so for this reaction, for example, the score is going to be what's represented at the bottom. I can stop briefly for any question about reaction expression scores. If there isn't, if you prefer to keep questions to the end, I can keep going.
It's as you prefer. Either way is okay.
Okay, just because it's a lot of information, so if somebody needs some clarification, I'm happy to stop and go through that.
Are there any questions for Sarah?
Okay.
I think everybody.
Okay, I'll keep going.
There is a question in the chat here, maybe really quick. There's a question from Quentin that says, can you define TMM? This is transcriptomic data.
Yeah, I always forget what it stands for, but it's just a normalization that we do to the counts. It's like the transcript, it's something equivalent to transcript per million, the TPM. So we can use TPM or we can use TMM, but it's just a normalization of transcript counts.
Thanks, he says.
Yeah. Okay, so once I have the reaction expression scores and to compute flux, so flux is equal to the catalytic rate times the abundance of the protein. But because we don't have the proteome, we use RAS as a proxy for the proteome or the reaction expression score. And then because it's really hard to find catalytic rates, There's a scarcity of those in databases, and it's just really hard to find them. You can use some tools for predictions, but we found a work that introduced the apparent catalytic rate. And so instead of using kcat times the abundance of the protein, we're using kap, which is the apparent catalytic rate times the reaction expression score to compute the biologically feasible flux. And what we mean by biologically feasible flux is really if I have like enough abundance of a certain protein, then what is the maximal or the biologically feasible the flux that can biologically that can feasibly go through that reaction. So we compute the apparent catalytic rate first using some other flux data. I can talk about the details offline because then we're going to get stuck just in there. So we compute kap, we compute reaction expression scores, and then we compute VBF, where VBF is just kap times reaction expression score. So VBF is what is going to tell the machine learning model how the fluxes should naturally look like. And then so this is how you can limit your predictions, or this is how you can direct your predictions towards a feasible solution for my simulation. Okay, now we have expression scores, we have the metabolic model, and we have the biologically feasible flux. So now we can look at how we use the machine learning model for simulation. So we compute VBF through the process that I talked about earlier, and then we take this as input for the machine learning. And so the machine learning will take biologically feasible flux, which is based on the transcriptome from context specific, that is specific to the context of that experiment. And then, so the neural network that is used here is just a typical dense neural network. It uses gradient descent to update its predictions. But what's different here is the loss function. We don't use an off-the-shelf loss function, but we use a custom loss function that integrates the differential equations from the modeling of the network. So so we create a mathematical modeling of the metabolic reconstruction with different constraints. Like, for example, I don't want I have some constraints on the uptake of certain nutrients, or I want it to be at steady state, or I don't want fluxes to be wild, so I don't want them to be greater than the biologically feasible flux. And I want fluxes to be positive, and so on and so forth. And as much as you can add as many constraints as you like. So if you want to simulate cofactor utilization within the model, you can come up with constraints that represent cofactor utilization, and you can add those to the loss function. I won't go into the details, but I'm happy to talk about it later on if anyone has any questions. And there's actually a work that has already been published by another group, and we're kind of enhancing, or we're starting off of that work and incrementing it with the different constraints and with the VBF. So I'm going to go through an example where we have some concrete data set that we've been applying to this pipeline. So that's from the Quantitative Plant Science Initiative that came out of Brookhaven National Lab. And it's a study of how zinc and iron affects the growth of plants. And so we're looking at how different treatments of zinc and iron affect the plastidial pathways. And so the experiment was conducted by growing sorghum and poplar plants under 5 different conditions: the control, limited zinc, limited iron, excess zinc, and excess iron. And then they took, our collaborators took samples from root and tissue in 5 time points, or at least we look at 5 time points here, day 2, day 4, day 7, 14, and 21. So if we apply reaction expression scores, this is an actual example from the metabolic model. And this is a reaction that can be catalyzed by either of these 3 enzymes, but it just so happened that each of these enzymes has only 1 subunit. So basically, the score for each enzyme is the sum of the abundances of the paralogs. And then the reaction expression score, here I compare the score under control growth conditions to the left, and then to the right, you can see limited iron condition. And so we can see here that already just by analyzing the enzyme complex, I can see that under limited iron, there's a large difference, or there's a huge difference between the score under limited iron and control condition. And so this also affects the biologically feasible flux under these two conditions. So under the control, reaction expression score is 297, and then VBF is 1000, because KAP was, I think, 3 point something. And then limited iron, it was 400, but then VBF was 1366. So you can already see how this code could affect the results of fluxes going through that reaction in simulation. We can also analyze the network as a whole just using reaction expression scores. And this is a scatter plot of RES of each reaction. So each dot here is a reaction. And so the x-axis is the score under limited zinc, and the y-axis is the score under control. And so any point that's to the left of the identity line would be a reaction that has a higher score under the control. And so it's we say it's downregulated under limited zinc. And then anything to the right is upregulated under limited zinc. And so the line plot will show you that on day 2 and 4, we had a lot more downregulated reactions. But then on day 7, we start getting to some kind of a steady state where it's similar thereafter. You can use this to just see how the whole network functions, but then we can also use it to compare different conditions, like comparing limited zinc to limited iron for the same species. But we can also compare across species because of the implied homology between these two. And as I mentioned, they both have the same network, so we can really see how each one of them behaves under different conditions. So for example, under limited iron, poplar has a lot more variability than sorghum. And we can also go to a more detailed analysis, and we can look at specific enzymes. And so here I'm giving the example of the central carbon metabolism. And so we can see comparing two species, for example, under limited zinc, sorghum and poplar have totally different reactions to limited zinc. And you can see specific enzymes, and you can go look into the details of these enzymes, see, for example, which genes were up or down regulated, and so on and so forth. And so finally, after analyzing reaction expression scores, we computed VBF for all the reactions, and we used what we call a sandbox model. So this part is still work in progress, and so we're using a very minimal model that has, I think, about 40-some reactions, just to have a better understanding of what's going on with the machine learning model. And we can go look at individual reactions as compared to if we were using a model with 400+ reactions. So this sandbox model was extracted from the sorghum model. And its biomass will only have one component, the 3-phosphoglycerate. And so here what we're trying to do is just biosynthesize 3-PG. So we run the machine learning model, and these are using data sets from different conditions. So we have the control, limited iron, limited zinc, excess iron, and excess zinc. And so this is the plot here shows the fluxes. So the y-axis is the flux, the predicted flux using the machine learning. So we can see that there's a variable carbon assimilation. And so with limited iron, it's almost in the 20s. And then with the other conditions, it was up in the 40s, or like, yeah, almost 40-some. And there's a variability in the assimilation of carbon. And because 3-PG has 3 carbons, So we expect that the assimilation of CO2 would be 3 times as much as the biomass, and that's what we got from the profile from the fluxes that the machine learning predicted. And so using this, we were able to just understand how the machine learning functions and also double check whether the results that we got made sense from just by following how the flux goes through different reactions and how they are interrelated. So the next step beyond using just the machine learning with the formulation with the FBA formulation, one could integrate secondary metabolite to the network. So what we've been using is just the primary metabolic model, but you can integrate secondary metabolite pathways within the primary metabolic reconstruction, and then use the machine learning to predict secondary metabolite production, for example, or identify key enzymes to improve the production of that secondary metabolite, or just whatever the objective is. You can add constraints to the machine learning model and help you understand how that secondary metabolite is being biosynthesized or how to improve that. And Sam is here. I don't know if he wants to talk about this part. It's about so Sam is working on this next step. Maybe he can give some more details.
Yeah, hi. Can you hear me okay?
Yes.
Yes, hello. Yeah, so it was actually a coincidence actually that when I logged into Zoom this morning, I realized that this talk was actually being hosted at the University of Minnesota. But we won a grant to actually implement this tool in KBase actually to create an app in KBase. Wherein if you actually were to train our machine learning approach on a data set for a second species that generate the same secondary metabolite and generate a trained model that actually would make predictions for how the secondary metabolite would, be biosynthesized or which enzymes in the primary metabolite network would actually, the expression of which would actually change the production of the secondary metabolite. So we actually just started work on that within the last year, and I am still looking for a computational postdoc that would actually come join Kathleen at the University of Minnesota to work on this. So if anybody talking knows somebody who might be finishing up their PhD or would be interested in this kind of work, please get in touch. But yeah, this will be an extension of what Sarah's been working on now. So I'm going to pass it back to Sarah. Thank you.
All right. Yeah, so Sam's email is down here and it's going to be on the last slide. So if whoever is interested, please shoot him an email. Okay, so with that, I'm just going to recap what I was saying, and then we can take some questions. So in this talk, we've talked about what's a pipeline that we've been working on to integrate omics data sets within the metabolic reconstruction. And so we've used implied homology to reconstruct the metabolic model of poplar and sorghum from the manual curation of Arabidopsis. And we've showed how we can use this along with just a transcriptome from experiments to generate hypotheses and compare different tissues, compare different treatments within the same species, but across different species. I also showed how we can use machine learning to enhance simulations, remove linearity, and the highly constrained flux balance analysis. And then it also allows us to add as many constraints as we want beyond the linear programming model. And so we can extend this to integrate secondary metabolites, like what Sam is going to be doing, or to incorporate cofactor binding, which is part of our next step for the QPSI project. So I'm just going to open it up for questions. And I would like to thank my collaborators. Sorry. We've had some really great collaborators from Brookhaven National Lab. They worked on the data set, and from Lawrence Berkeley National Lab.
Fantastic. Thank you, Sarah.
Yeah,
Are there
sure.
questions for Sarah? Feel free to unmute yourself and ask, or you're welcome to put your question in the chat as well.
Hi, Sarah.
Hello.
I'll go for the first one. So, okay, I am not a Flux or a machine learning person, but I'm going to ask you this.
Yes.
Sometimes the transcriptome shows that a specific gene, enzymatic gene, is downregulated, but then that information actually is just that expression is going down, but the protein level, tends to stay the same. And again, this list is really big, right? Does that affect the model if you're just using the transcriptome data? Does it make sense what I'm asking?
Yeah. That's actually a great question. It's part of what I was talking about earlier in terms of there's assumptions behind the reaction expression score. And the first one is that we can use the transcriptome to approximate the proteome, actually, because we don't have a proteome, but the proteome is really what tells you what's going on with the protein itself. But because it's really hard to get a proteome, we use a transcriptome. But going back to your question, I'm just going to take this example here. And so it's true that one of these paralogs could change significantly, and it could also not change at all. So for example, if you look at this little green one here, it was kind of expressed here, but it's really not expressed in the limited iron. But then you can still see that it didn't really have an effect because we sum up the expression of all paralogs. And then we take the minimum across subunits, and then we sum up again the expression of the enzyme. So that kind of alleviate that effect of one gene being highly expressed or not expressed at all under certain condition, affecting the whole score of the reaction. So even though that specific enzyme became 0 under limited iron, you can still see that limited iron score is much higher than the control condition. I'm not sure if that answers your question, but
yeah, it partially answers the question. Yeah.
If you need more clarifications, I can try, but also
no, that's okay. I
Sam
mean, like,
can help here.
I'm also probably not going to completely understand it. That's good enough.
Okay.
Other questions for Sarah?
Oh, I have another follow-up question, if that's okay.
Sure, go ahead.
So I'm assuming that the answer is yes, but I still want to clarify this. So you have used micronutrients, and I'm assuming that the similar logic could be applied for many other cofactors like coenzymes or glutathione, ascorbate, and things like that, right?
Yeah, so the thing is, I didn't talk about micronutrients here, but it was part of the pipeline. The only thing that you need to, in order to fully integrate that, what you need to do is to actually know which enzymes bind which micronutrients. So with iron and zinc, it's really hard to find that information in databases because experimentally, it's hard to know, I guess, or it's expensive.
Yeah.
So we've been finding it really difficult to find information online, but we've tried some tools like predictors or people have come up with different, like there's a AlphaFill, which is an equivalent of AlphaFold, but AlphaFill predicts which enzymes bind which cofactors. And so as long as you have that information and you can express it as a certain type of constraint, in the metabolic network, you can definitely include it in the machine learning model. So
Thank
you can, yeah, you can have a
Yeah.
constraint like if there's limited zinc, then this is how that reflect in a mathematical equation. This is how it reflects on the flux, for example. So then you can incorporate it with the machine learning model.
Awesome. Thank you.
Mm-hmm.
Maybe I think Shen is next.
Okay.
Oh, thank you. So Sarah, I'm just, can you elaborate a little bit more on like how the machine learning can improve the problem for the FBA analysis? Because you mentioned the FBA is limited to like linear reactions.
Right.
Can you elaborate a little bit more on that?
Oh, so the FBA is limited to linear equations. So it can only, so if you take the metabolic network and you try to express it as a mathematical model, you can only use linear equations if you're using FBA. So here you can see that, for example, I can only say whatever the steady state is, whatever the stoichiometric matrix times the fluxes is equal to 0. Or you can say MC has to be less than some upper bound. Let's say this is oxygen, MC1 is oxygen. It cannot be more than this certain uptake. Or you can just say the total flux going through reactions that produce a metabolite cannot exceed the total flux going through reactions that produce that same metabolite. So I want it to be steady state, but I can only use linear equations. And so if you have anything other than linear equations, it becomes something other than FBA. And so any change to any of these fluxes that I have here in the flux vector will be linearly reflected into the biomass. And so it doesn't allow for that much variability and that much freedom in the constraints. So, but with the machine learning, I don't have to stick to linear equations. I could use quadratic equations in a mixture with linear equations, and it can be a really mixed mathematical model, and it will still work because in the end, all you need are those differential equations that you can tell the neural network, this is how much error you have in satisfying these constraints, go do better. And so it comes up with another prediction, and you say, oh, now this prediction is much better, so the error is less. You're going on the right track, keep it up. And so it comes up with the next, it just iterates over its predictions, and you give it feedback based on how well the predicted flux is actually satisfying those constraints. And you can just add as many constraints as you like. You can change them, you can enhance on the constraints that you have. And so that gives you a lot more freedom with how much you can add to this mathematical representation of the problem that you're trying to solve.
So am I understanding this right? So the first part is the FBA analysis, and then the like whatever the result coming from the FBA analysis, like get fed into the neural network and for the machine learning part. You
don't have to go through FBA. We're just currently, because our representation or our model looks pretty much like FBA, just so that we can see how the machine learning is working right now. But you don't have to go through FBA at all if we don't have to. So let's say you have a fluxome, you have a transcriptome, or you have a transcriptome and metabolome, you can directly use your machine learning with any of the constraints that you'd have come up with for the loss function.
Okay, thank you.
Yeah, sure.
Michael, do you have a question?
Yeah, thank you very much for the talk. Now, I'm also not somebody that dabbles in modeling in any degree to this that you presented here, but I just had some questions specifically because I'm interested in the capacity of this to make maybe novel predictions in terms of secondary metabolism, and like maybe, you know, optimizing transient or heterologous expression of genes in another plant. So the first is, you mentioned that it goes into this Arabidopsis annotation database, and then it kind of, from the transcriptome, it takes everything from Arabidopsis. So I am assuming that it takes like kind of all the kinetic and flex data that's been associated with the genes and the orthologs that are within Arabidopsis, and it assumes all the other plants that it's modeling against, or basically just different Arabidopsises, to say it simply, right?
Yeah, so I might have not explained it properly, but I'm going to attempt an answer, but Sam can chime in. So Arabidopsis is only used to annotate the genome with gene functions and associating genes with pathways.
Okay.
That's the only thing that Arabidopsis is used for, but Sam can take it from here. Sam, you're muted.
Sorry, I was muted. The Arabidopsis being the model organism for which many of the enzymes have been characterized was the source of our enzymatic functions. So we actually spent a couple of years manually correlating a set of over 1,000 reactions with the Arabidopsis genes associated with them. And from then, when it was there to actually annotate other genomes, we have to be very strict how we define the function, the enzymatic function, and how they are associated with the biochemical reaction. Because for at least the FBA to work, the biochemical structures themselves have to be determined. We can't use abstract biochemical reactions or very loosely defined enzymatic characteristics and so on. And so that's why we created our own database to do this. But having done that, yes, we annotate other genomes, and we work with the assumption that the primary metabolite network is highly conserved, which almost all of it is, across flowering plants, at least across flowering plants, But, obviously with specialized metabolism, it's not. And so we wanted to actually integrate some additional pathways that are specific for sorghum, for example. We would actually go and add those. But yes, Arabidopsis is our starting point.
Okay, yeah. So kind of building on that, I just kind of wanted to ask from a lay perspective, what kind of information do you need when you are inputting this secondary metabolic network also and adding on to like branching off of primary metabolism? Do you need simply enzyme function? Do you need more detailed information to make novel predictions or to optimize the system for production of a certain metabolite? Do you need kinetic data? What are the constraints and what are the things that you need to input into this model in order to make it make a novel prediction rather than just model what is already observed?
You want the gene to be characterized, first of all, and I understand that, with specialized metabolisms, you will have some in the pathway, but you will also get gaps in the pathway. And you do need, I'm talking about gaps because you do need a complete pathway. You do need the carbon and the nitrogen and the other element to actually be able to go down every step in the pathway to the end products from the primary metabolic precursors to the end product. So if it's a long pathway, for example, and you have gaps in it, you're going to get stuck.
Yeah.
You're going to get stuck at the first intermediate that you can connect to the primary metabolic network. Each of the biochemical reactions should have biochemical structures, not just an abstract name for a compound, but an understanding of what the actual biochemistry is, so that we can actually balance the flow of the carbon and the other elements through the network. And then finally, you do need the genes perfectly from a genome. For a plant species, let's say perfectly from a genome, because if you use somehow characterized transcripts from a transcriptome of a plant for which the genome hasn't been sequenced, what will be a big problem is that you won't have consistent identifiers or protein sequences. for that enzyme if you wanted to find the ortholos in further species, right? So that, but all of that is basically just bioinformatics, it's underlying bioinformatics problems that we have to cover. But at the end of the day, if you have a fully characterized pathway, it should be straightforward to integrate with the primary metabolic network.
I'll just add one
Okay.
point for the kinetics. You mentioned earlier that we could use whether we were asking if we use kinetics for Arabidopsis for other species. We actually don't do that. So we look in databases for Arabidopsis KCAT values, for example, but we also look for poplar and sorghum. And we've been using the prediction tools to try and predict based on the sequence of the protein or the protein sequence to predict the kinetic rate. But we stopped at some point. Or Sam can also add anything if there's
Thank you.
any-
Quentin, go ahead. Oh, yeah.
Hi, this is a really cool talk. And I wanted to hear more about the back end and like some of the measurements on either not necessarily metabolism, but some of the data that's guiding your FBA constraint functions. You're probably measuring growth, but like what are some of the other constraints? How are those defined? And if you could wish for more constraints or data to support that, what would that data be?
So are you talking about these specific constraints that we use here for the loss function?
Yeah,
Or-
like what are the fluxes that are constraining here, and did those come from the Brookhaven data?
Oh, so VBF is what came from the data set from QPSI project. So, and that could be replaced by any data set really. Ideally, and like the really perfect situation would be to have a metabolome and a transcriptome, because the transcriptome is what tells the machine learning this is, so VBF is just telling the predictor this is the biologically feasible flux. Anything you predict beyond this is not biologically feasible, so you're going like off chart. And anything lower than that is what we kind of expect with this expression set, with this expression profile of the genes. But ideally, and now here I'm using this machine learning method as a solver. It could be used as a predictor or as a solver. As a predictor, you tell it, it's like the typical machine learning where you have a training set where you say, this is a face, predict whether it's a face, and I tell you whether it's actually a face. But here, we don't have that information of what you're predicting, whether it's a face or not. You can just predict, and if you're within those constraints, then you're right. If you're off constraints, then you're wrong. So I just try to make the error go to 0, and if it goes to 0, then it's on the right path. So then ideally, if you have a fluxome, for example, and a proteome, then you can use the proteome to guide the predictions, and you can use the fluxome to actually really double check if those fluxes are correct or not. So if what you're predicted is equal to what I expect to see, then you're really doing a great job. So ideally, we want a proteome and a fluxome, or a metabolome, or a transcriptome and a metabolome. But because it's hard to get like a pair of these, we're happy with just getting a transcriptome, better even the proteome. And then we can use the constraints to guide the predictions and say, well, yes, if you're satisfying all the constraints, then you're doing a great job. Then I like those predictions.
Okay, so the
Actually,
data
if you
that's
don't
in your,
mind, sorry.
or go ahead.
I'd like to add a little more to that, actually, because I think it's actually really interesting. The people that published this original paper who worked with Bill, they looked at E. coli, they looked like a bacterium, and they were actually able to control the media that the bacterium was growing in and measure directly how fast the bacterium was growing. And we can't do that. We can't really, I mean, they had very specific time frames that they're working with, and we can't do that. But I've been talking to my collaborators on this project with Brookhaven, and I think one of the best things that we've got that we've actually started to talk about integrating is data on gas exchange and data on chlorophyll. And so they actually did some spectroscopic measures of the leaves that gives you an idea of how metabolically active the leaf is based on the greenness, the chlorophyll in the leaf. And because we actually have photosynthesis working in our metabolic model, we actually have the electron transport chain that's absorbing photons and transferring electrons. We can actually integrate that on some level to say, okay, this is how much carbon dioxide it's consuming, and this is how active the photosynthesis transport chain is. And so we just started to talk about that. We can't go inside the leaf and just take a sample of all the fluxes that are happening at the same time, but we actually can use some proxy data to try and get an idea of what's going on. So I think it's really interesting.
Yeah, I think that kind of helps to answer my question. So this was more constrained by what's metabolically feasible, unless like you weren't taking some CO2 rates or oxygen exchange rates or biomass growth rates. That's for future iterations.
Right.
Okay. Thanks so much. Really good talk.
Thank you.
Yeah, thank you again for an excellent talk and a great series of questions. This has been a very interesting discussion, and I think there's some compliments in the chat. So thank you again, Sarah, and thank you everyone for your questions. Thank you, Sam. And yeah, please stay tuned. In another couple of weeks, we've got another talk, and we hope to see you all then.
Yeah, thank you all very much. And please feel free to email us if you have any further questions, or if you want to try some parts of this that are already fully functional.
Excellent.
Thank you.
Thank you.
Thank you.
See you later, everyone. Have a good weekend.
Yeah, thank you.
Bye-bye.